of Evolution and Ecology, also on the 5th floor, like Bruce. Sadly, we can't claim him as a Canadian, but he's going to be talking
from Boston.
Yeah, it's pretty close. Lower Canada. He's going to be talking about next gen sequencing techniques and sort of the implications for phylogenetics.
Yeah, so normally for this class, I talk about what you would call phylogenomics, which is integrating evolutionary reconstructions and genome analysis in a variety of ways. And this year, they forbid me from talking about that. So what they wanted me to do, which I'm going to try to do, and please stop me anywhere along the way, is give you sort of an outline of sequencing methodology. And no one can keep up. I mean, there are like 20 new companies that are in the wings about to announce new sequencing technologies. You just have to follow the press to really stay along. But I'll give a little bit of sort of the history for people who can see the progress. So before I moved to UC Davis, I was at a place called TIGER, the Institute for Genomic Research, which was the place that sequenced the first genome of any organism in 1995. I wasn't there when they did it, but when I moved there, we had about 100 fluorescent automated sequencers. That was the cool thing, and now a single, $100,000 desktop machine outstrips all the capacity that we had back in 1998. So it's been completely crazy. And I don't know what, are most people here, how many people here actually have a significant amount of experience using or generating next-gen sequencing data? So not too many, okay. All right, so everything I'm going to talk about is in essence fluff relative to this slide. So the key thing in all the sequencing technologies you need to think about is maybe about 5 to 10 different parameters related to that sequencing technology. And really, it is impossible to keep up with the developments. Everybody thinks they're going to make $1 billion off of human genotyping in some way. And there are a huge number of companies out there developing technologies, and really, you can't keep up. So, oh, man. I got to pay more attention to these things. Obviously, cost per base pair is a really important thing for determining, exactly how you're going to apply a sequencing technology to a particular area. The read length, which I'll talk about a little bit, is highly variable between the different technologies. And for certain purposes, short reads are fine, like resequencing genomes. If you already have a reference genome that's high quality, you can get short reads from a new sample and map them to the reference genome. Or you can get short cDNA sequences to do a transcriptome sequence if you have a reference genome. But for many purposes, including de novo sequencing of new organisms, sequencing from environmental samples, or trying to construct haplotype structures, longer reads are really beneficial. So the read length is really important for the uses of the sequencing technology. There's something that still keeps this old terminology, although it's not always used in exactly the way it was used originally, called paired end sequencing. The terminology comes from sequencing from the two ends of a plasmid vector, in essence. And there are a lot of different ways that people apply something like this to try and read sort of pieces of DNA that are a set unit of distance apart in the reference DNA sample. And when you constrain that set distance, you can use the paired end sequence data to do mapping of some kind or genome assembly. A really important thing that is coming out now is that we are really, in essence, not limited by sequencing per se. We're limited by being able to feed samples into the sequencing machines. So if you want to do population genetics, one Illumina sequencing run is probably enough sequencing capacity for almost every project in the world. Mixing together samples and tagging them in some way and feeding them into one sequencing run is the big challenge that people face now. How can you put together 100,000 samples into a single run of the machine? Because for many studies, you don't really need more than a few thousand or tens of thousands of sequences per sample. What you need are to be able to collect tens of thousands of samples and feed them into the machine. Of course, depending on your purpose, the error profile in the sequence is in the method is really important. So if you want to do comparisons of closely related organisms to do population genetics or something to that effect, the SNP error rate in the sequencing method is really important. If you want to do something like deep phylogenetics and you're getting a sample from the first representative of a family or an order, the sequencing error probability distribution is not that as important as for doing closely related organisms. And then this ease of feeding is related to this barcoding potential. The different sequencing methods, some of them are better able to pool together samples and tag them in some way. It's generally called barcoding, where you add a small piece of DNA where you know the sequence of that piece of DNA, and then when you read the sequences at the end, you can figure out what sample that sequence came from. So if you look at the sort of history of DNA sequencing, I assume most people know some of this history. So in 1977, the Sanger sequencing method was described. This is the standard method for DNA sequencing that was used up until about 2004. There was an alternative DNA sequencing method that some people used called Maxim-Gilbert sequencing. Sanger eventually took over, and most people, again, up until 2004, and still a little bit today, use the Sanger sequencing method. And then, for many, many years, this Sanger sequencing method was continually refined. And what I'm going to talk about when I go through sort of the history is that the biggest transition was automation of the Sanger sequencing method with fluorescent DNA sequencing methods. And that sort of revolutionized genomics and sequencing and set the stage for a lot of the next generation sequencing methods. And then in the last few years, there's been this crazy progression of sequencing technology. So in 2005, the 454 sequencer came out as a commercial product. There are lots of sequencers that have come out but have never been sold. We're not counting those. If someone just announces that they've sequenced something in their garage and don't actually sell a piece of equipment or release the data, we're not going to count those. So then in 2006, a company called Solexa put out a DNA sequencing system. It was rapidly bought by Illumina. 454 was bought by Roche in 2007, and in 2007, Applied Biosystems, the big sort of gorilla in the sequencing world, put out this ABI Solid sequencing method. And these were the three sort of next-gen sequencing methods that were available for a while. And now in the last year or two, there have been these sort of third, fourth, fifth generation DNA sequencing technology, and I'll talk about these a little bit right at the end. What I'm going to focus on mostly is these, what are generally called the second generation sequencing technology, although already, really, I'll just get to the punchline. Already, really, the 454 and ABI Solid technology, you can just sort of throw them in the trash compared to the revolutions that have happened with the Illumina sequencing method. And then Illumina has sort of won the battle, in essence, for these second generation sequencers. And now the question is one of the third generation sequencing systems going to replace the Illumina method? So I'll go back. This is how I learned to do sequencing. So the Sanger sequencing method is pretty straightforward. You take a DNA sample, you use a DNA polymerase in the test tube, you add a primer at the beginning of your sequence. So if you know the sequence, you can use a primer from inside the sequence. Alternatively, if you don't know the sequence, you would clone your pieces of DNA into a plasmid vector, then make lots of copies of that plasmid by growing it in E. coli, and use the plasmid sequence as your primer for the sequencing reaction. And then the way the sequencing works is you have 4 different tubes. Each tube, you add a terminator of the sequencing reaction that terminates a small fraction of the sequencing reactions whenever an A is put in tube number 1, and a small fraction of the sequencing reactions when a C is put in tube number 2, and in tube number 3 for G, and tube number 4 for whatever one I didn't say already, A maybe. And so you have these 4 different tubes in essence, and each one of them will end up with pieces of DNA that stop at the point where there is an A in the A tube, in the newly replicated piece of DNA that the DNA polymerase is copying. And then you run these out on a gel, separate them by size, and you get this DNA ladder. And you can just read the ladder corresponding to the sequence from the original organism. And this was the way it worked for sequencing for 10 or so years. And it was painstaking and slow. Some people developed semi-automated methods to read these X-rays, to scan in an image of this and try and read the DNA sequence off of those X-rays. But the real revolution came in the late 1980s, early 1990s, when Lee Hood and a few other people invented an automated sequencing system that used fluorescent dyes for each of those letters. For A, C, T, and G, got a different colored fluor. And in one tube now, you can run the reaction for all the different nucleotides, and then separate the DNA by size with little capillary tubes, and then read the sequence coming out with a laser beam. So you separate the DNA by size, and you read at a particular set point the color that is coming through the capillary. And then you get a little plot, an electropherogram, that tells you what the sequence is. So this was completely revolutionary. It allowed people to massively scale up DNA sequencing. And this is what led to the genomics revolution. So the speed at which people could sequence with this technology was so much greater than had been done previously that people felt like there was a revolution by doing this. And what people did for many years was apply this automated sequencing method to a variety of biological sort of problems. And eventually, everybody became rate limited at the time in feeding these machines. So again, I worked at the Institute for Genomic Research. We had 100 plus of these machines. Each one cost, $700,000 or something to that effect. And each one could read originally 96 sequences at a time. It had 96 little tubes, and then 384 sequences at a time. And you had to have a different plasmid prep for each one of the sequences that you were running on this machine, and a different DNA prep of those plasmids for each of these reactions. So what we would do, and what we mainly worked on, was automation of this colony picking. So you would take a DNA sample, shred it, put it into a plasmid vector, ligate the ends, and then electroporate that library into E. coli. Plate those E. coli strains out on typical E. coli growth plates. And now you'd get a few hundred colonies on one plate. We had, as we called them then, massively parallel undergraduates, picking colonies with toothpicks. Imagine picking 200,000 colonies with different toothpicks over and over and over again. It was really a great job. So robotics was really important for revolutionizing the sequencing approaches at the time, by allowing people to no longer have to pick colonies themselves, and you'd have a robot. So there were a lot of robots that were developed that would take a picture of these plates of E. coli colonies. Then software would tell this automated picker where to go pick up a single colony and drop it off into a little growth microtiter dish. And then you could have an automated robot, which other companies sold or developed, for doing DNA preps from those colonies that were grown in these microtiter dishes. And eventually, sequencing got what we thought was really cheap, which was down to maybe 50 cents per sequence that came out of this type of protocol. And this sort of, revolutionized the world of biology by allowing for high throughput sequencing. Most of this was done with what's called shotgun sequencing, where you take a DNA sample, you shred it up, and you just randomly sequence pieces of DNA that come out of that library at the other end. And then you use computational methods to compare the pieces to each other. For example, if you're sequencing a genome, to try and stitch back together the genome sequence of an organism by looking for overlaps between the different pieces. If a genome was relatively simple without a lot of repetitive DNA, this worked well. If the genome was more complicated, this didn't work that well. So the first genome was finished in 1995, and there was a rapid progression of at least attempts to sequence genomes using this DNA shotgun sequencing method. And then eventually people even applied this automated Sanger sequencing to environmental samples. So this is what I work on, where you go to a field, you collect a soil sample, you extract total DNA from that sample, and you randomly sequence that DNA from a collection of organisms. It's called metagenomics or environmental genomics. And this was, again, originally done with the shotgun sequencing with the Sanger method. So this was the way it stood until really 2005, 2004, and 2005. And so when I was at Tiger, I went in 1998. My first genome project, I sequenced a bacterial genome. The total cost of the project for a 2 million base pair bacterial genome was about $800,000. So, and today the cost is about 50. So, I mean, it's just the sequencing technology with the next generation sequencing methods has really been completely insane in terms of cost. So that's it for the history before getting to the next generation sequencing methods. I just want to stop here and ask, does anybody have any questions about sort of this? Nobody really does this anymore. You probably have a sequencing facility somewhere where they'll do a little bit of sequencing with one of these Sanger sequencing methods. At UC Davis, it costs between $3 and $5 total to get a clone sequenced. We can, with Illumina sequencing, $5 buys you, about 100,000 reads. So, if you think about a way to make use of the Illumina sequencing technology, you're going to be much more cost effective in doing something with DNA sequencing. So what happened was all these previous methods, and they were all revolved around basically the Sanger sequencing technology related to clone-based sequencing. So you had to take the DNA, put it into E. coli, grow up lots of copies of those E. coli's, and then sequence off of those clones. And again, the automation of colony picking and clone preparation and DNA preparation was a key part of this whole process. And what these three sequencing technologies changed was the way they work is instead of cloning, you physically make clusters of amplified product off of single DNA molecules. So you take your DNA from your sample, you do some library prep on that sample, and I'll tell you about the different ways that these work. And in each of these methods, what you're doing is you're physically isolating a single piece of DNA and then making lots of copies of that piece of DNA. So that has now circumvented growing something in E. coli. And you make millions of copies of each little piece of DNA, and that then allows you to run some sequencing reaction and get enough signal that you can read the answer by having 100,000 or 1 million or more copies of each piece of DNA. And that's what each of these methods, the revolution that they provided was really figuring out ways to do this cluster development in the absence of DNA cloning. I'll post all of these. There's a good review paper on all the methods up to Illumina sequencing, basically, that Elaine Mardis wrote, and I'll send this around. She's at the Washington University Genome Center and helped test and or develop many of these methods, and this is a really good review paper. So this is a summary slide of these three methods. So the 454 method, 454 was bought by Roche, so some people call this Roche, some people call it 454. The technology is something called pyrosequencing, so it will be called any of those three things, Roche, 454, or pyrosequencing. Illumina bought Solexa. Most people don't refer to it as Solexa sequencing anymore, and just call it Illumina sequencing, and it's also something called sequencing by synthesis. That's the way the sequencing works. And then the Applied Biosystems has this ABI Solid method. And they basically, they all go through, you take DNA, you shred up the DNA with some specialized method to chop up the DNA into the right size pieces. You add little adapters onto your DNA. Depending on the method you're using, the exact method of adding adapters varies. And then for the 454 and the ABI Solid method, basically what you do is you encapsulate each little piece of DNA from your library in what's called an emulsion product. So you basically make little mini beads that encapsulate the piece of DNA. And you run a PCR reaction inside each of those beads. So it's called Emulsion PCR. And this makes lots of copies of a target piece of DNA inside a little tiny microbead, say of agarose. There are different chemicals that people use, but agarose is one of them. And then after you make those millions of copies, you do something with these beads that allows you to run a sequencing reaction on those beads. And so the Roche 454 method does pyrosequencing. The ABI Solid method does something called sequencing by ligation. The ion Torrent method, which many of you may have heard of, does another type of sequencing off of these emulsion PCR methods, the emulsion PCR products. Illumina is completely different. It doesn't do this emulsification. It actually spots the DNA basically onto a little glass slide and runs this little thing called basically bridge PCR on the glass slide, and it makes millions of copies physically around your little piece of DNA on the slide. So you get what are called clusters on the slide of copies of the target piece of DNA. And then when you run the reaction, you have to remember which position you are at when you run the sequencing, in essence, cycling. And you figure out, base number 1, base number 2 by their coordinates on the slide of these clusters. And you identify where the clusters are by the first few cycles of the reaction. But again, they all work on the same principle, making lots of copies in some physically constrained environment, and then running a sequencing reaction and reading the sequence as it comes off, with some fluorescent or electrochemical or other method that's reading the sequence of bases in each of those little collections of DNA pieces. So this is one of the Roche systems. So Roche was the first one out the door with this and did temporarily rule the market because there was no other choice if you wanted to do high throughput, next generation sequencing by some method. It again uses this pyrosequencing method, which has been around for a little while, but was not automated into this emulsion PCR method until the development of the Roche system. So the way it works is basically you have a DNA polymerase, it incorporates nucleotides, and pyrophosphate comes off while it's running the DNA synthesis reaction. And through a series of chemical steps, the reaction basically converts this pyrophosphate into light. And then you have a light meter basically reading the reaction as it's happening. And there are these 3 main steps. You have library prep, and then this emulsion PCR, and then a DNA sequencing reaction. I don't know if people actually care about the exact details, but I'm happy to tell people about it for all these. They're all really cool. I mean, they're just completely brilliant technical tour de forces of how people figured out how to get some cloning independent cluster generation and sequencing to work. So with 454, what they do is you take your DNA, you shred it. You can shred it into a variety of sizes. The size that you shred it into determines, in essence, how far apart the 2 ends of the piece of DNA that you have are. And then you ligate onto the ends adapters that correspond to specific things that are going to be used later in the reaction. So you run this in vitro ligation reaction. And then I think they've modified this a little bit now, but the original method, this is from Elaine Mardis's paper, basically what you're trying to do is physically isolate single-stranded pieces of DNA that have one or the other of these adapters, depending on the orientation of them. And then you take those single-stranded pieces and you isolate them with magnetic beads or some type of biotin labeling and magnetic beads, although there are a variety of derivatives of this. And then you encapsulate them in this emulsion product. So you do this, you try and, in essence, estimate how much DNA you have, and then you try and generate these emulsion products such that there's a single piece of DNA. You do basically a Poisson statistical expectation such that most of your little emulsion beads have 1 piece of DNA in them. Not 2, then you'll have a big problem with your sequencing, and not 0, then you're going to waste money. So a huge challenge in all of the emulsion PCR-based methods is to try and get 1 piece of DNA in each of the emulsion beads and detect that and not run sequencing reactions afterwards that either read 2 DNA sequences at a time, which you can't interpret, or have no DNA in them. And this is one of the biggest sources of failure of these methods, like the Roche sequencing method. Frequently, the emulsion PCR step and the bead counting and the bead sort of working is the problem step. And so you run this PCR inside these little microbeads. You purify them and prep them in some way, and then you take these microbeads now that have hopefully millions of copies of your original piece of DNA in each little bead, and you put them into this flow cell. And so there's a little, basically massively tiny microtiter dishes that have space for one of these beads to fall into. And you try and pack enough of these beads into this flow cell such that you'll be able to read a lot of DNA sequences. So the latest Roche machine has about 1 million of these spots that you can read about 1 million sequences if it all works well. If you don't do it very well, you can read a lot fewer sequencing reactions. And then once that's done, you run the pyrosequencing reaction itself in each of these beads. And you're basically flowing chemicals across your little flow cell and watching the light that comes off with this luciferase reaction. And so you basically have a microscope that's watching as the reaction happens. And you get a pattern. And one of the biggest challenges with pyrosequencing is that if you have a run of, say, 3 A's that are going to be put in your DNA sequence, the method will just, the DNA polymerase will just put in A's. You just feed it the chemicals. It doesn't have a terminator. You're just giving it enough A's to feed into the reaction. And if there's a run of A's, it will put them all in. And the only way to tell how many A's it put in is by the amplitude of the light that you're getting off, basically. And so the most common sequencing error with the Roche 454 method is misestimating the number of polynucleotides you have in a run. Does that make sense why that happens? And that's a really big deal for any of the methods that use a chemistry where the DNA reaction is allowed to keep running as long as it can for a particular nucleotide. And not being able to tell how many are put in is a common problem with many of these methods. So the Roche equipment has evolved quite a bit. The original machine was called a GS20, and then an FLX machine. And then now there are 2 different versions of this FLX titanium. And one thing that Roche did, which all the other companies are trying to do too, is they made a mini version of their machine that does, it's like mini-me, it does 100,000 sequences instead of 1 million sequences. And it's a benchtop. We actually have one in my lab. It's smaller than most PCR machines. And the cost per reagents is, it does 1/10 as many sequences for 1/8 the cost, or 1/5 the cost. I mean, it's more expensive per base pair, but it's a little more manageable in terms of the total amount of work and the amount of data that's coming out of the system. And I want to throw the machine away. But that we can get to at a later point. Are there any questions about the Roche methodology? There are a lot of other places that have developed pyrosequencing. There's a QIAGEN pyrosequencer now that some people are using for epigenomics to look at methylation status. And there, I think, are a couple of other companies that are now marketing pyrosequencing-based sequencing methods. Yeah?
Just wondering how you have these plates with all these microscopic wells in them. How do you clean the machine? Like, contamination is a big problem with PCR-based stuff.
Yeah, so... Yeah, you have a new cell for each time you run it, and you're, in theory, supposed to run the emulsion PCR phase in a separate place from where you run the sequencing phase. So we have a separate little workstation to run the emulsion PCR. In the end, if you're not working with forensic type samples, a lot of the time you have milligrams of DNA, right? So the contamination is not as big an issue for many sort of genome scale projects. But when you're moving into PCR amplification of your DNA samples, so a lot of these methods are also being used for ribosomal RNA sequencing from environmental samples of microbes. And then you're running a PCR reaction to even generate your initial sample of DNA, and then you run an emulsion PCR to get even more of each of the ribosomal RNA samples. So when you go through multiple amplification steps, you run even more risks of contamination. So there are some issues with it, but it's much more of a problem that it just fails. And Rob Knight, who works on microbial sequencing, has a great slide where he says 454 is the temperature at which your money burns. Yeah.
Basically, the same sample in all the wells, right? With all the each plate is one sample.
So what you do is you take your DNA, you shred it up into pieces, and you, let's say you have 1 million different pieces of DNA from your original sample. Each of those ends up in an emulsion reaction. And then those get amplified. And now you have a pool of emulsion beads. Each one is a different piece of DNA, hopefully. And then you just pour those all into the little slide. And you run that all at once. Now, the Roche machine actually has different chambers. So you can add a little gasket onto it and separate them. If you had 8 different samples, you could know where they were on the little dish. The little mini machine has it's just 100,000 sequences all mixed together. What people do now is they tag each sample. If you wanted to pool 100 samples into a single run, you can add, when you add ligate on your adapters, you can add an extra few bases to it that will tag your sample with, say, 10 nucleotides or 4 nucleotides that correspond to sample 1, and 4 other nucleotides that correspond to sample 2. And then when you pool everything together, you just figure it out bioinformatically afterwards which came from which sample. It can be very challenging to pool those together in the right relative abundance to get equal number of reads from each sample. That's a fine-tuning that is very hard to get to work. Does that make sense? So again, the Illumina and ABI Solid methods, they're the same principle. The details are very different, but the principle is completely identical. Make a lot of copies in physical isolation of your non-cloned piece of DNA and then run some sequencing reaction. So the Illumina also has gone through many different generations. They also have a mini machine called a MiSeq now. The HiSeq machine can generate, 700 gigabases of DNA per run or something like this. It's completely ridiculous. So the way the Illumina works is basically you do this thing called cluster generation, which I'll tell you a little bit more about in a minute, and then you run this sequencing by synthesis reaction. So again, the mechanism is different. It's not doing pyrosequencing. It's doing sequencing by synthesis. It's not doing emulsion PCR. It's doing these clusters. But the general principle is the same. You take your DNA, you add adapters onto the ends. You can modify these adapters to add on barcodes if you want to, or a variety of other things to do different types of gimmicks with your DNA sequencing. And what you do is really, it's completely cool. You basically have a slide, and on that slide you've spotted some PCR primers that correspond to the adapters that you've ligated onto the end of your DNA pieces. And you then, in essence, covalently link your DNA onto the slide in random spots all throughout this slide. So you have this little slide. It's overloaded with PCR primers. You then attach your DNA sample, sort of string it out like single-stranded pieces of your DNA onto this slide. So you have these little pillars that correspond to your DNA sticking out in this sea of PCR primers, basically. And then you run this thing called bridge PCR, or bridge amplification, where in essence, the details don't really matter that much. In essence, what you're doing is you're making copies of your piece of DNA using the local primers that are physically around that piece of DNA. And you get these little bridge products where the piece of DNA is extended to another primer, and you run this PCR basically in physical isolation as opposed to on these emulsion products. And eventually you get these clusters where one region of the plate contains lots of copies of that original piece of DNA. Now, attached to the plate. It's completely brilliant, and really quite cool. And now all you have to do is figure out a way now to run a DNA sequencing reaction that runs on these clusters, and you can watch this with a microscope or with some other methodology, and they tried a bunch of different methods, but the one they use is basically a nice microscope. And you run this sequencing by synthesis. The exact details don't really matter, but you get fluorescently labeled tags for each nucleotide that's being read on each of these clusters. So again, just like with the Roche method, you don't want to have too much DNA on your slide. If you have two different pieces of DNA in one region, you're going to get a cluster that includes multiple types of DNA, and then you won't be able to understand what the data that's coming off of that cluster is because it will be reading A, T at the same time from the different pieces of DNA. You then run multiple cycles of this sequencing by synthesis reaction and watch with the sort of video microscope as the different colors come in different regions of the slide where these clusters are. And then there's a lot of informatics that has to figure out where a cluster actually is. And it, I think, iterates over the first few cycles to figure out where the clusters are and then can focus in on those regions. In the original development of the Illumina method, this cycling reaction and the sort of scanning the slide and finding the clusters was very slow. So you could generate about 10 nucleotides a day, maybe, with the machine. So if you wanted 50 base pair reads, it would take 5 days to run the machine. If you wanted 100 base pair reads, again, you're getting into weeks of time on the machine to just run it to get longer reads. It turns out that the method originally as it was developed couldn't read much longer than 90 or so bases anyway, but even those 90 bases took weeks to generate in many cases. So this is an example of one of the flow cells for the Illumina machine, and in essence, same general principle as most of the other methods. And you run, however many cycles you can run. You get this phase problem with many of these methods, where if, as you get later and later in the cycles, if something has gotten sort of out of phase with some of your pieces of DNA, they're now going to be, like the 99th base might be overlapping and being read with the 100th base. And you get some challenges as you run out longer and longer reads with many of the machines. The sequencing by synthesis is kind of cool, but the exact details don't really matter for anybody's evolutionary purposes. I'd be happy to talk to people about it. One of the great things about Illumina sequencing, which works a lot better than the 454 sequencing, is the ability to make libraries that you can then read the two different ends of this paired end sequencing. So the way it works with the Illumina sequencing is pretty simple. You take your DNA pieces, you add your adapter tags, and then you run a sequencing reaction. And in essence, you can wash off everything, all the reagents off the plate, and run the sequencing reaction with the other primer, the other adapter, and now you're reading the sequence, in essence, in reverse. And so if you have a 300 base pair insert, you can read 100 base pairs on one side and 100 base pairs on the other, and now get paired end sequences that are 300 base pairs apart, but you read the left and right 100 base pairs. The new DNA sequencing chemistry for Illumina, they claim, can get up to 250 base pair reads. So you can now imagine doing a 500 base pair insert and reading both ends and getting overlaps of that. Paired end technology is really important if you can figure out a way to do it for like a 10 kilobase piece of DNA or a 100 kilobase piece of DNA. If you can read the ends and you know that they're 10,000 base pairs apart, that can be really important for building haplotype structures or assembling genomes. Yeah?
Does the length of the insert affect the bridge implementation?
Yeah, so when you start with the Illumina DNA, you take your DNA sample and you shred it up into pieces. And what you try and do is you target pieces that will work for that bridge reaction as well as for some of the other steps. And the ideal is about 300 base pairs, it seems. You can't go much bigger than that, at least. I mean, people play with the chemistry all the time to figure out other ways to do it. But if you had, imagine you have a 20,000 base pair piece of DNA. It's flopping around on your slide surface. It doesn't bend over and find the primers that are nearby it. It can reach over to some other spot, and the reaction doesn't work. You don't read the sequence very well. So most of the time, people do about 100 to 300 base pair pieces of DNA. And if you wanted to do a 10,000 base pair insert, you have to figure out a way to cut out the middle and then attach the two ends together and then feed it into the Illumina system and run the bridge reaction where you've sort of chopped out the middle 9,000 bases of your piece of DNA. And there are tricks people have developed for doing this that work much better for Illumina sequencing than work much better for 454 sequencing than for many of the other methods. So I misspoke before. 454 sequencing method, one of the things people still use it for is that the large insert libraries work a little bit better than with Illumina sequencing. Yeah.
Is that mated pair?
Yeah, so mate pair. Yeah. So, and I'm not going to go into the chemistry, but I have some slides on this if you want to learn about it, and it's in this review paper. Again, the ABI Solid method is yet another one of these methods to do making lots of copies of a single piece of DNA and then reading the sequence in some way. It does an emulsion PCR step, but then its sequencing method is different than the sequencing method used by pyrosequencing, and I'm not going to go into all the details. So when Elaine Martis wrote this review paper, and even today, you can compare the methods, and these sort of rules sort of still hold. So if you compare the three different methods that we've talked about here, ABI Solid was generating the shortest read lengths. Illumina was next, and Roche was next. That's still sort of the case. So Roche has now pushed the sequencing limit to about 1,000 base pairs, at least in their lab. And they're releasing chemistry for people to do this. It hasn't worked perfectly well for a lot of people, but in theory, people will be able to get 800 base pair reads with Roche sequencing. So for many purposes, read length is all that matters. Of the three methods we've talked about, the 454 sequencing is the best for that. The runtime, so Roche takes you a couple days to make the libraries and to do the emulsion PCR, but the run of the machine only takes a few hours. Whereas if you're trying to do long reads with Illumina sequencing, it can take days, and so can the ABI Solid. The total amount of DNA you get per day and the total amount of DNA you get per dollar is a lot better for the Illumina sequencing than the other methods. And this was the old Illumina sequencing when people were only getting about 30 to 90 bases of DNA sequencing out of it. The new Illumina sequencing is by far and away sort of the winner in terms of cost relative to these other systems. So if your goal is to generate a lot of sequence data, figuring out how to do something with the Illumina machine is probably your best bet, even today, even though we'll talk about some of the fourth generation sequencing methods. Each of the methods has advantages and disadvantages in terms of the sort of accuracy, the ability to make large insert libraries, the ability to do different types of chemistry, different types of sequencing. Some work well with PCR products. So originally, Roche was sort of the place where people went if they wanted to PCR amplify something and then sequence it, like HIV sequences from a quasi-species or ribosomal RNA sequences from an environmental sample. The other methods have now basically caught up, so they have developed ways to take PCR products and adapt them and feed them into the sequencing systems pretty well. And now, for example, for microbial studies, for ribosomal RNA sequencing in microbial studies, most people have shifted to doing Illumina sequencing of PCR amplification of ribosomal RNA rather than 454 sequencing. Even with the longer reads that you get with 454, the higher quality with Illumina and the sheer volume you get with Illumina, people have shifted over to that. Is there a question? So here is sort of the cost per million base pairs. The Roche was much higher. This was again in 2008. The Illumina was much, at least an order of magnitude lower. ABI Solid has been basically in parallel to Illumina, but just sort of got swept up by not coming out soon enough and not having a lot of people develop different methods to feed it. And so there were 5,000 papers using Illumina sequencing to do a variety of things with all the tricks needed to make libraries and to do the informatics and to feed it. And there were correspondingly 50 papers with ABI Solid. So it may be equivalent in many ways, but it just lost out in essence in a race. Yeah?
Is it true that it has higher error Illumina as well?
It has higher what?
Higher error?
Well, so the people who, for certain applications, the error rate was not so bad with ABI Solid. But again, I think the development of Illumina, so many more people had Illumina machines and that played with the informatics and the chemistry that now Illumina is significantly better with sort of the error distribution. But it's not like an inherent, it may not be an inherent issue as much as just lack of development with the method, whereas pyrosequencing, it always is going to have an issue with the homopolymer runs, really no matter what you do. So that's my opinion, is that Illumina is the winner in all of this. So for example, I just got this off the Illumina website, the new HiSeq 2500 machine. If you run it in its long run, which takes 11 days, you can get 600 gigabases of sequence off of a single run. You can, however, run this machine fast. It's about a day. You get a little bit less total sequence, but you still get 120 gigabases of sequence in a day off of this machine. This is what, for example, the big genome centers are buying a bunch of these HiSeq 2500 machines to run the crank on, say, a sequencing of a human genomes, for example. Where if you wanted to sequence one human genome that's 3 gigabases, you frequently need about 100x coverage to get good calling of SNPs in a genome with these sequencing methods. So one run of the HiSeq 2500 is, 200x coverage of a human genome. And that's about right for getting good calls of individual bases. So does that make sense? What you're doing is basically all of the methods have a reasonably high error rate for individual sequences. And for sequencing a genome, if you get 200-fold coverage of the genome, that is, on average, each piece of the genome has been read 200 times, you can now make up for the errors by having depth of coverage for particular regions of the genome. That only works if the error frequency is random. So if the sequencing method is highly biased, you will get the same error over and over and over again, no matter what your coverage in a particular region. So what you really want is a sequencing method that has a knowable and predictable error distribution, and hopefully is close to random. None of them are really random, but some of them that have been developed are more biased than others. But again, this is now getting, you can do the paired end sequencing with 150 base pairs for each sequence with the Illumina. So that's getting, that's 300 base pairs basically for a single piece of DNA. Compared to when they started at 36, that's, a 10-fold increase in just like 3 years of development. And again, they have a little mini machine called a MiSeq. This costs about a little more than $100,000. And for $100,000, you can get basically, in 1.5 days, 6 gigabases of sequence from that machine. And the sequencing runs are much cheaper because you're using fewer reagents. So it's, maybe $1,000 or $2,000 for a run here, total reagent costs. So, $1,000 for 6 to 7 gigabases of sequence. You're overwhelmed with data here. So again, the key that everybody has to deal with now is if you're not sequencing, say, entire genomes to do comparisons of entire genomes, is figuring out how to pool together samples tagged them in some way and mixed them together into a single run of one of these machines. Because you don't need, however many, I forget how many sequences this is actually generating for 6 gigabases, but 100 million sequences or whatever it is, you don't need that for to do population genetics of a mouse. You don't need 100 million sequences from each mouse or each plant sample. And you might want, say, 10,000 sequences from each. So if you're getting hundreds of millions of sequences in a run and all you need is 10,000 from each of your samples, you have to basically figure out a way to pool together thousands of samples into a single run and tag each of those with a little barcode and sort them out afterwards. And so there are a lot of companies out there that have developed methods to try and help people do this. Most of them are 2 or 3 years behind in the profile. So like Raindance has this technology that can help you do, with very controlled conditions, about 24 or 48 samples at once. And that's great, but we're talking about, for microbial sequencing, doing 1,000 in a single run. So we'd have to do 50 runs with this machine just to prepare 1,000 samples. So it's not keeping up really with the technology. So other people are developing microfluidic systems to try and make barcoded libraries with a microfluidic device that might allow you to do thousands of samples instead of tens of samples. And again, there are Raindance and Nimblegen and Agilent and a lot of these other companies have developed technology to try and help people pool together samples and feed them into one of these sequencing platforms. PCR actually is a pretty good strategy if you can figure out how to feed these machines, because you can, in some of them, you can add the barcoded adapters onto the PCR primers after you run the PCR. So you don't have to synthesize 1,000 different PCR primers. You can just have your set of tags, ligate them onto your PCR products, and then clone those, and you might be able to do this in a massively high throughput manner. Yeah.
What about like AFLP or something for this as a way to chop things up before?
You mean to try and identify a region of the genome to do, or?
Well, no, it's just you're talking about like ways so that you could potentially do more samples in one run.
So the AFLP might be a way of screening samples, right?
Yeah.
We don't need to screen samples, we just need to sequence them. So I mean, the sequencing is so cheap that it's more expensive literally to do PCR for samples than it is to sequence many of them. So if you develop some way of like saying, I'm going to screen my 10,000 samples to decide which ones to do, which that's what I'm guessing you're sort of suggesting, but maybe you're suggesting something different. So if we have methods that are RIFLEP-based methods that we used to use to screen samples to decide which ones were variable, that is more expensive than just sequencing, that if we can pool them together.
So what, in like about 4 slides back, what are the runs actually, the prices per run actually based on?
So I'll just tell you about the mini machine. So the mini machine, it's about $1,000 to $2,000 in reagents for a full run. The HiSeq machine is probably $20,000 for the big machine. It has 4 flow cells. I mean, it's basically increased its capacity by adding extra sides to the flow cells and adding additional flow cells. So you still have to use a lot of reagents to run those machines, and the cells themselves cost a lot, but it's I think it's something like $20,000 for one of the HiSeq runs, and you're getting, 1 billion sequence reads.
If the cells are imprinted with the primers, how are they reused if at all?
You don't reuse them, you throw them away. Maybe some people try and use them, but we don't. Yeah?
I think Mercedes is sort of asking about the genome reduction strategies that most people know as RADseq, which is just AFLPs, which is cheaper than doing whole genome because you can get, thousands of homologous loci from hundreds of individuals in a single run.
Yeah, so that's what the Agilent SureSelect system is basically doing. So there are a large number of kits and companies that have tried to develop methods like that will feed into now Illumina sequencing. The one that a lot of places use is this Agilent SureSelect system, which is basically a column. Agilent is basically a primer printing company. So they have a big dot matrix printer that prints primers. And they can print a gazillion primers corresponding to target regions of a genome. And you make a column, basically, that you flow your DNA against, and it captures the DNA that corresponds to that region. And then you wash everything else off, you flow off the captured DNA, and feed that into a library construction system. And so that may be, sort of the moral equivalent of some of these other methods. But the challenge is you have to pair that to making Illumina libraries. So it has to be set up such that you can, in a high throughput manner, feed your SureSelect products into making Illumina libraries. So now Agilent sells robots for like $1 million that pair up with the SureSelect system that will make your library for you after, can make 96 libraries in a day, basically, or something to that effect. Is that more what you were sort of referring to, like target enrichment, or it doesn't matter?
Well, we're doing definitely like we're doing more like it's not as automated as high throughput as what you're talking about, but you can sort of purchase your own primers and make your own tags and use your own AFLPs to more inexpensively generate large samples of homologous sequences from large numbers of individuals.
Yeah, I mean, you can do it with PCR, right? I mean, if you have a conserved enough region between organisms, and most of it, what you pick to do depends on whether or not you need haplotype structure recovered for some organisms. So if PCR primers across a region loses the haplotype structure. So some of these, there's a company that Agilent just bought that has some technology that allows you to capture 100,000 base pair pieces and retain the haplotype structure when you're making the library. So you, in essence, shred up 100,000 base pair pieces into a sub-library and then do the sequencing off of that so that if you care about haplotype structure, you can recover it. But I mean, there are 10,000 of these approaches. The key is to being able to take advantage, to me, I think, the key is to be able to take advantage of the incredible decrease in cost of sequencing without making your workload go up too much in terms of prepping samples. So there's no doubt that right now sequencing for many projects is cheaper than sample prep for projects. Because this, all the machinations that you do with sample prep and PCR and pooling together samples and extracting DNA and tracking them and doing voucher specimens and all these other things, the sequencing is now the cheap part. And the sequencing is getting cheaper at a rate faster than Moore's law. So I would expect in a year that you can, in essence, consider sequencing to be the cheapest part of your project. It's the dealing with samples and dealing with the bioinformatics that's going to be the challenging part. It's not. I mean, obviously, if you're doing, large genomes of organisms and you need hundreds of those, sequencing is still going to be an expensive part. But if you need 10,000 base pairs from 10,000 organisms, that's one HiSeq run. Not even. You get more than that. So I can't even keep up with the methods, but one method that I would call generation 3.5 is the ion torrent method, which is kind of great for the coolness factor, and it's not clear how useful it is yet, but it's pretty friggin' cool. So it's basically 454 sequencing in essence. You can imagine it's completely like 454 sequencing. It was developed by the guy who invented 454 sequencing. And instead of doing pyrosequencing and reading things with light, it's reading things by electrical charge. And it can do the equivalent amount of sequencing in 15 minutes or so, as opposed to in a whole day. Now, it's not very accurate, and it's still a pain in the butt to feed this machine, but it is, I know people who have these machines, and they're really quite cool, and if you are able to make libraries quickly, you can get data by the end of the day. I mean, it is really quite a remarkable thing. It's not very accurate right now, so you have to sort of use it in the context of what its error probability is. It has some of the same problems with homopolymer runs as 454 sequencing. And what's really, I consider the fourth generation of sequencing are what are called single molecule sequencing methods. So all the methods I've talked about so far generate some type of cluster or collection of copies of an individual piece of DNA. There are now these DNA sequencing methods that actually sequence single pieces of DNA. The one that I assume many of you have heard of is Pacific Biosciences. It basically affixes a piece of DNA into this little well. They have a special name for all of this, and I don't even want to go through the names that they have for it. And it attaches the piece of DNA to a DNA polymerase, and it watches in real time as that DNA polymerase copies the piece of DNA. And it reads this with some type of fluorescence, but it's literally watching a single DNA polymerase enzyme copying a single piece of DNA. And it watches this is in DNA polymerase time, which is 15 bases a second, basically. So it can do 1,000 bases in 15 minutes. And if you can jam a long enough piece of DNA into this well attached to the polymerase, in theory, this method could read 100,000 base pair pieces of DNA. Right now, people are using it to read anywhere up to maybe 5,000 base pair pieces of DNA. You can get it to work for maybe 10,000. It doesn't seem, I don't know anybody who's gotten it to really work well for longer pieces. It has an incredibly poor accuracy. Just does not, the method is very, very inaccurate, but if you need scaffolding or large pieces of DNA for some purpose, again, like assembling genomes or doing haplotype structure or some other purpose, this is a very good complement to Illumina sequencing. So you use Illumina sequencing to get coverage and PacBio sequencing to get large pieces of DNA. You correct the errors in the PacBio sequence from the Illumina data, and you use the PacBio data to get your scaffold for a genome of interest. And it is, I mean, if you can prep the library, you can literally get data in 15 minutes. If you can figure out how to analyze it, you could run a new reaction based upon the initial result and in essence do real-time experimental studies where your results change what you're going to do with the sequencing an hour later. It is completely amazing. We have one at UC Davis. It's not been used in a lot of studies effectively yet, again, because the error rate is so poor. But it's still really cool. And one thing that's really important about this method, it's watching in real time as this polymerase copies DNA. You can actually use it instead of for sequencing for studies of enzymology of polymerases. And a lot of people are starting to use the PacBio system for studying the kinetics of different enzymes. You can also use it to look at modified bases. So methylation slows down the DNA polymerase. And so epigenetics, you can use the timing of the sequence output to tell you whether or not some piece of DNA was methylated or damaged with, say, UV radiation. People apparently are also using it to do RNA, single RNA sequencing. So there are lots of cool adaptations to the PacBio machine that are not just sort of sequencing per se. And that is probably why a lot of people are sort of playing around with it, even though the sequence data that is coming off of it is not anything to write home about. And it's still kind of cool, but I'm going to just skip over this. There's a company called Complete Genomics that has a method, because I want to get to this last one, which, if this actually works, we should all buy stock in this company. So this Oxford Nanopore company has been around for a long time, and there are many companies that have been around for a long time talking about this thing called nanopore sequencing. And basically, there are a lot of different derivatives of it, but you have some pore. What Oxford Nanopore actually does is they use a real protein that has like a hole in the middle of the protein, one of these membrane transporters. And they measure sort of the electrochemical gradient and current generated through a membrane by stuff, in essence, any type of thing. People have been using nanopores for all sorts of different purposes. And what they've been trying to figure out for many years is how to adapt this technology to do DNA sequencing. And what they've managed to figure out, at least they claimed this at this Marco Island genome meeting about a month ago, is a way to, more accurately than they've been able to do, to use these nanopores to track a DNA sequencing reaction and to track, in essence, the charge and electrochemical changes that happen during a DNA sequencing reaction. Here's a little bit more detail of their model where you have some sort of nanopore, and I think this is a DNA polymerase, and then a piece of DNA, and you're threading the DNA through the nanopore and running sequencing, some type of synthesis reaction or DNA copying reaction as it goes through. And this is them, after playing around with it for a long time, they've found that a G has this signature with the current, a T has this signature with the current, a C has this signature, and don't know, here's A up at the top. So each of the different bases have a different signature in terms of the current that's going through this nanopore. And they can read single molecule sequencing with this nanopore sequencing. Now, who cares? This is kind of boring until you get to the next part, which is this is what they announced. This is their sequencer. It is a thumb drive that can do DNA sequencing in the field and download the data to your laptop. All self-contained within this, it's not, a tiny thumb drive, but that's ridiculous. It's not clear how well this works. It's not clear, if the error rate goes up a lot when you attach something to a thumb drive system as opposed to a more controlled environment. But I fucking want. They're just, I'm a field microbiologist. I mean, if I could take this out on a deep sea research cruise or up into the Sierras or whatever, I don't know. I mean, what they didn't announce and what I've asked them and I still haven't found out is what you feed into this. Like, I mean, who cares if you can have a thumb drive sequencer if you need 70 ultracentrifuges to prep the DNA? So it's unclear exactly what you need to do to feed this machine, if you call that a machine. But there are a few other companies that have been trying to do field DNA sequencers like this. Again, I don't know what the DNA preps for most of them are going to be, but this is single molecule sequencing, very fast, reasonably high throughput. None of the methods that are out there have even come close to the Illumina throughput right now. So for a while at least, if you want to get deep coverage of something, or if you want to count things like RNA, Illumina sequencing is still going to be a great place to do this because the cost per read is still incredibly low for Illumina sequencing. So for example, doing a transcriptome with Illumina sequencing, where you're using the number of copies that you get for a particular RNA to tell you how many to do gene expression levels, you need to do millions and millions of sequences to get a good estimate of the relative abundance with a broad dynamic range across rare and abundant transcripts. Illumina is still going to win for that because of the sheer number of sequences that you get per dollar. So, this thing may win out in the coolness factor and in speed and turnover time. And the other, if this works, there's no startup cost. You buy this, it's $900. You don't need a $500,000 sequencing machine. That's it. You're done, is their claim. So this is the first real democratization of sequencing, if it works. Because even the Illumina MiSeq machine is $150,000. The Ion Torrent is $90,000. And again, for each of those, you need another $200,000 in equipment to really make libraries well. But $1,000 that is completely self-contained, disposable, high-throughput DNA sequencer. Pretty amazing.